But AI and synthesis allows us to do this completely different. Here we have a completely unconstrained genotypic space. Synthesis is template free. We can synthesize whatever we want. And now, with the ability to start speaking DNA, I think it will be very interesting to see how much we can, basically navigate.
Welcome to Galaxy Balance, a podcast exploring the intersection of cutting-edge science, technology, and humanity's long-term future. Each episode features visionary thinkers working on transformative technologies, including synthetic biology, AI-driven science, longevity, and genome engineering. Today's guest is Dr. Marc Güell, a synthetic biologist working at the frontier of genome engineering, AI-driven biological design, and translational therapeutics. His work focuses on developing new ways to read and rewrite genomes with high precision, including programmable integration systems, AI-designed biological tools that go beyond what evolution has produced. In this conversation, we'll explore how generative AI is changing the way biological systems are designed, what it takes to translate powerful genome writing technologies into real therapies, and how Marc thinks about the future of synthetic biology as both a scientific and engineering discipline. We'll also discuss the founding of Integrate Therapeutics, the challenge of building deep tech biotech companies, and how imagination, including science fiction, shape the way we envision and build the future of life. Marc, welcome to Galaxy Balance.
Oh, it's a pleasure to be here.
Yeah, thanks for coming on. I'd like to open up just a bit more about you, about your arc and your story, and what led you to this point where you're at now. So if you could share what got you into science and what led you on this path.
Yeah, I guess, I don't know, it's a bit, so somehow I think as a kid I was always saying that I wanted to be an inventor. I'm not sure what this means, but well, at the end I have always been, I think, more interested in what is building things. I really enjoy reading about descriptive science, but definitely when it comes to the first person, what I really enjoy very much is to tinker and build. I grew up in an area which is very surrounded by nature. I grew up in the Pyrenees, these mountains between France and Spain, in an area which is surrounded by volcanoes, actually. And all summers I was sent to the coast with my parents, where the Pyrenees, with my grandparents, where the Pyrenees die into the sea. And there I was also very exposed to nature, right? Chasing octopuses, clams, fish, starfish, and all of that. So I had, I would say, a very, let's say, rural and peaceful childhood. And when I turned 18, I moved to Barcelona to do college. I studied undergrads of organic chemistry and chemical engineering, but very quickly I got very attracted to let's say build using biology. It's true that my first jobs were still as organic chemist, which I enjoyed very much. Because this is this very experimental, radical experimentation, right, with these big syntheses and things. But yeah, for the PhD, I moved to Heidelberg. I joined Luis Serrano Lab at the MBL. And basically, we were working on a large European project where the big goal was to build what my boss was calling it a living pill. So basically, we were using as a chassis as Mycoplasma pneumonia, one of the simplest bacteria that exists, to really first understand this bacteria as much as we could, but also equip it for different applications. But on that moment, it was very clear that my fascination was technology, because I remember when I was doing presentations, part of my PhD was to characterize the transcriptome of this microbe, to then use this knowledge for engineering purposes. But my boss was always joking that I spent 90% of my slides when I was presenting my work in talking about the techniques, and then one slide to present the results. So yeah, somehow it was clear that my interest was in the technology, and then it was very clear that also for postdoc, probably the Church Lab at Harvard would be an ideal place. And yeah, in 2019, I moved to Boston to join George's lab. And yeah, it was a so fantastic time. It's one of these decisions that I would never change. Sometimes I wonder, what if I had studied maths, or what if I had studied medicine or whatever instead of, but definitely joining the Church Lab was really something that I would never change. Yeah, I think George influenced me in a way that I like very much, right? Always this vision beyond horizon. I loved one of probably the only rule that existed in the Church Lab, which was not using the word impossible. which sounds metaphoric, but I think it's very dangerous to use the word impossible, especially in these times in biology. So during that time, I work in different projects. I work in both prokaryotic engineering as well as mammalian engineering. So I did genome recoding in E. coli. So we built this genome with 57 columns. Also was the time of CRISPR, right? The big boom of CRISPR. So we used CRISPR to engineer pigs for genotransplantation purposes. Helped to start the Genesis Bio, which also was a fantastic experience. And I think it's one of these incredible examples of how far can synthetic biology go, right? With this recent basically first groundbreaking transplantation speak to human during the last months. Which even, basically, it seems that we are successful in engineering things that are so deep to biology as cross-species compatibility. And also from that time was very nice also the college, right? Like you, Cory, so many people, right, that I worked together, Luhan Yang, Christian Mali, Lily Ostrov. And this was until end of 2017. Then I went back to Spain to start our current lab, the Symbio lab in Pompeu Fabra University in Barcelona, where we mainly focus on exploring molecular paradigms to engineer biology. We have been, let's say, working in three main blocks. One is chip writing, that probably we'll talk a bit later. Also, we are very interested in let's say, the foundational paradigms of engineering biology, what we call synthetic evolution, or this convergence between AI and synthetic biology. And another topic that we are applying synthetic biology since quite a long time is skin microbiomes. We have been also very interested in using the microbes to engineer the skin. Have been doing this since more than 10 years, a project that started kind of garage biology, so with a friend from grad school, Bernard Petzel and Veronica Udova, where basically started more than 10 years ago a company called Biomedic, where basically in the beginning we were just trying to see, can we change the skin microbiome. We got some small funding in Chile because it was quite hard to get funding for this type of science. We could build the first IP, the first demonstration that we could truly change the microbiome, the skin microbiome. And then we came back to Europe, where we could get, let's say, more traditional investors like Johnson & Johnson and later Beiersdorf, and really establish, in this case, Cutibacterium magnes, one of our more common skin microbes, as I think a great chassis to do engineering in the skin. So we did design of natural probiotics, which seems to be very effective for acne and other well-being. And this was had several clinical testing, and even later on, Beiersdorf acquired the company, and I think they will soon announce some interesting outcomes of these skin microbiome-based health solutions. We're still in the lab. We are super interested in Cutibacterium magnes. Now in the lab, we use it more, let's say, as a truly synthetic biology chassis, where we build different genetic circuits to modulate different things from the skin, from, let's say, sebum secretion, inflammation. Also, we implement sensing on these microbes. Given that this bacteria is quite special, since it lives quite deep in the skin, it has the advantage first that it engrafts very well. So we can deploy to the skin and it stays for a long time. We have shown that it can stay for weeks. And then also, given that it's so deep, it can interact quite efficiently with human skin and physiological centers, like, for example, sebum secretion, inflammation, or even other crazy applications, like even using them to generate thermogenesis. So this more or less is our what we do in the Symbio lab in Barcelona. So engineering paradigms for biology, three main blocks: chip writing, paradigms of synthetic evolution, and the skin microbiome.
Excellent. Thanks for that summary. And I appreciate your story of sharing that your thesis advisor said you'd spend all your slides on the technology and one on the biology. Reminds me of myself. But really, think of the key technology advances allow us to get to this point. Like X-ray crystallography, we can get the structure of DNA. DNA sequencing, we can identify, hey, repeat elements. These might bind the other viruses. This is the CRISPR system. So it really is advancing with these key technologies. What technologies do you think are up and coming that are really going to change synthetic biology in the future?
Yeah, I think it's a very special time, right? We have had, let's say, different exponential kind of revolutions, right? From the sequencing revolution in the first decade of 2000, the synthesis revolution in the second decade, where, yeah, whole chromosomes were synthesized from both bacteria and even some eukaryotic chromosomes. And this has been incredible, right? A lot of progress has been done, big milestones, like as you were saying, right? CRISPR to correct mutations in humans, reprogramming the immune system to fight cancer, autoimmune disease, or even what I mentioned before, right? Xenotransplantation, even using basically, yeah, this engineering biology to alter something as deep as the across-species compatibility. But all of this was done in a way that it's very much by inspiration from nature, right? Looking at nature and somehow refactoring or transplanting parts. But since we don't speak DNA, it was hard to do something truly synthetic, right? And I think now it's a very exciting moment, right? Because not only we can still read and write and edit, but now we can start speaking DNA with these new AI tools like LLMs, which I think it's a big foundational change for synthetic biology, and I think we are seeing a lot of applications. First in the protein domain, but now we start to see pieces of genomes, genetic circuits, which I think it's interesting, not only because of these practical applications that start to come up, but also I think there is something very inspiring, because we are all fascinated by biology, right? We see these incredible properties, right? Animals that resist cosmic radiation, like tardigrades, animals that almost never get cancer, like blue whales, etc., or animals that they have this extreme capacity to regenerate, and all of this has been generated by this, let's say, imperfect DNA replication, right? So there's a highly constrained genotypic space because it always needs to be close to the parental DNA, right? It's always based the template-based replication. And then, of course, so mistakes in this template-based replication and followed by selection. But AI and synthesis allows us to do this completely different. Here we have a completely unconstrained genotypic space. Synthesis is template-free. We can synthesize whatever we want. And now, with the ability to start speaking DNA, I think it will be very interesting to see how much we can basically navigate this way more unconstrained genotypic space. And another fundamental thing, which I find very, very inspiring, is the fact that up to now, always when a new genotype emerged, was independent on the phenotype, right? When the polymerase makes a mistake, doesn't know about the phenotype that will come after. But this, again, changes completely with these conversions between AI and synthetic biology, because we have methods to, for example, apply reinforced learning or other types of learning to the generative AI elements, which then we are doing a coupling of genotype generation with phenotype, which, I think it's another, very interesting property for exploring synthetic purposes. So I'm very, 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 very excited about the future.
Yeah, I think these are incredible tools, as you say, to speak DNA. That at least we can talk with the machine that can speak DNA. I talked with George recently, and he was very proud of the work Colossal did for de-extinction. Not just for the preservation or nature or de-extinction aspect, but as a tool to test genetic variants into phenotype. You could take these species, and we know all the differences between mammoth and the elephant, but then to predict which ones, good to predict, but actually to have this biological system to test that difference. So, as you were hinting at, what systems do you see to really high throughput this, of not constrained by small genotypic changes, but really explore the whole automated space, and then it could be at cells or organisms. How do you then test the function you want?
Yeah, that's a very interesting point, because especially most of the AI environments where these algorithms emerged, normally there is, let's say, more accessibility to data and an easier generation of tests. Of course, in biology, we need the hardware. So it's true that every time we are more efficient in multiplexing the synthesis of DNA with, for example, DNA chips, but it's true that still synthesis of large fragments, it's a clear bottleneck. It's difficult still to build, let's say, 100 or 1,000 5 kilobase pieces of DNA. So I think it's still probably a bottleneck compared to other, let's say, areas of applications of AI. So I think here, I don't know, there are different options. I think one option is the self-read systems that maybe they will enable us to, we have seen some examples of very interesting coupling of self-read systems with AI. And of course, maybe one day, really learning how to generate this template-free synthesis, maybe even, let's say, more coupled to biological systems. But yeah, so I think this is probably now is one of the biggest bottlenecks, right? How to couple this throughput that we have on the AI side with the hardware.
Yeah, and one interesting thing that happened with sequencing the human genome, the several key discoveries stand out to me. One, the junk DNA or dark matter, that most of it wasn't proteins. That was a big surprise. And a lot of scientists expected humans to have more genes, more than 100,000 plus, but the number was closer to mice and some single-celled organisms. But one of the things that we really discovered was the complexity with which humans can splice DNA. How many different splice forms and the control between that. Do you know of any AI systems or thoughts on how to investigate that type of genetic space?
Yeah, here I'm very fascinated about, let's say, I think how complexity emerges, right? For example, here, I think a small number of people could predict that LLMs had this kind of disruptive moment, right? Not many people who were betting on LLMs, let's say 20 years ago. So I think here we clearly we're missing some familiarity of computational tools to model these complex behaviors. But now I think here has been a very humble learning that maybe sometimes some of the algorithms or the formal paradigms that we are working with actually have a very big capacity to generate complexity. We are still learning what you just mentioned, right? How relatively or basically how we underestimated maybe the number of genes that can generate this very complex life being. Here, I'm not sure what will be the most adequate mathematics. Now we managed to kind of model the language of biology, but how to model, to put this into 3D. I think this is kind of an next frontier. How we will be able to model a cell? Now we model pretty well a genome or a gene, which has been amazing, right? To have this LLM that can start to speak DNA, but I think it will be very interesting how we manage to put this in 3 dimensions.
Yeah, there's lots of approaches to model a virtual cell. We've talked of some of Craig Venter's work and the synthetic genomes. They published a paper probably 10 years ago now on a virtual cell. I mean, you can argue what aspects they actually modeled, but now a lot of approaches, particularly at the ARC Institute, are trying to model it from the transcriptomics point of view. Where do you see the role and future of virtual cells or simulating life or entire cells?
Well, I think that for sure will be very interesting. Actually, many times when we were talking about throughput of to couple AI with hardware, I think that the big problem is that we don't have a good oracle for many things. So it would be fantastic to have more oracles. Now we have a small number, right? We have an oracle of structures, which is AlphaFold. We have some oracles of certain, let's say, properties like DNA binding for zinc fingers, for some limited set of proteins. But I think it's transformative to have these oracles because then we can jump into throughput of computing. And this, of course, when it comes to more complex systems, eventually the human body, I think this is something that for sure will be key in accelerating science. And here I think, because, for example, when we go to modeling, I think we have had a very important change of paradigm. We were maybe using more differential equations or even stochastic equations, some kind of paradigms that, they have been very useful for certain contexts. But for example, for AI, the mathematical paradigm has changed completely. Actually, the approaches that were competing several decades ago now are completely different. And this, let's say, neural network architecture has demonstrated that can really produce this complex and emergent behavior. So I'm very curious about which mathematical paradigms will be successful in modeling these highly complex systems.
Yeah, really interesting. And what like you say, it's difficult to predict. So few people would have predicted. I had a previous guest say it's even pointless to predict the long-term future because so many things come up. But like you said, one of the inspirations of George was seeing that long-term vision. I think even if it might not work out that exact way, it is important to model and look out to that future. And there may be some disruptive technology that totally changes it, but I think probably like you, my mind changed with having his over the horizon view of where you're going. And I really think that empowers scientists. So within this modeling and AI field, what is your lab doing to really bring about some new disruptive changes to the way proteins are designed, for example?
Yeah, so for example, we have been very interested in these conversions between AI and synthetic biology. For example, in the specific problem of gene writing, so we have been working on a specific molecular paradigm, which is combining the precision of CRISPR and the gene transfer capacity of transposons to build what we call a gene writer, or well, the field kind of calls it a gene writer. We have done different things to improve the efficiency of the system, to, especially in this case, very focused to medical applications, right? We have good programs for T-cell engineering, for liver in vivo gene writing. And yeah, we have done a lot of molecular tinkering, especially in coupling the CRISPR system with the transposase, so that to work well together. And we have very low off target, we have good efficiency of this large gene transfer or gene sequence transfer. And especially recently, we tried to really first do some bioprospecting, so to see if we could improve the parts that we are using. Again, this is let's say, a molecular paradigm that has two parts, a transposase and a CRISPR. So we have been doing the bioprospecting to find better transposases. Actually, we did search in all the genomes. We did find better transposases than the ones that we were using. Actually, we found about 10,000 systems that they have good, that they show good data. But probably some years ago, the project would have finished here as a reasonably successful experiment that you find maybe several tens of clearly better transposases than the ones we had. But now I think we could go a bit beyond. So now these 10,000 new transposases, we can use them to train, in this case, a protein large language model, and somehow then expand this biodiversity, because now we are not limited to these 10,000 proteins, but now we can generate pretty much as we want, because it's the computer generating them. And then, yeah, we started ranking the best synthetic AI-generated transposases, and actually we could find that now all our best transposases are basically AI-generated, so they do not exist in nature, which was a very interesting lesson. And probably the first time in my career that we have made, despite being called synthetic biology for many years, now that we have made truly synthetic parts that, in this case, perform and outpace what we find in the, let's say, the normal biology. So yeah, that was quite exciting. And this is exploring this part of using generative AI to expand the genotypic network. And now what we are trying to do is to couple this with different, let's say, tools that can bias more and more this genotype generation, especially using reinforced learning, and also using other elements, like, for example, chain of thought. So different tools that can help navigating this genotype network and influence it as much as possible to go towards the function that we want. This is a kind of a paradigm that we have repeated with multiple paradigms, with different molecular systems, with DNA transposons. We have also been doing it with retrotransposons, also we are exploring as RNA transposon, retrotransposons as an alternative methodology to do gene writing. And in both cases, it worked very well. So we are quite excited about, basically, what we were saying before, not only being able to read and write, but also start to speak DNA in a decently controlled way, right? Starting to write basic poetry in DNA.
Yeah, excellent point. Well said. And very exciting on the bioprospecting part. So did you actually sequence new bacteria, or did you just take stuff that was already available in public databases?
Yeah, to be honest, we didn't sequence any extra base. So we just used public databases. In this case, we focused on mammalian transposons, because in this case, we're very interested in gene writers for humans. Yeah. So we just search all the eukaryotic genomes available. and found a huge amount of unexplored,
That's exciting.
similar prototypes.
Craig Venter did this years ago, that he went into the ocean and started sequencing the ocean. And one surprising thing was how much of it was unknown. The availability of biodiversity is so great on this planet. So I imagine this work, if you had even more data, even more eukaryotes that could provide this, you could get even better models. But interesting you say you're getting the best ones are all AI generated. I'm interested, how different are they than the native transposons? Are they 5% difference, 10% different, or totally different? And
they are quite different. I have to say that this depends a bit on the cluster that we are modeling. So different clusters, we have seen more flexibility, in others less. But for example, our best piggyback transposon, which is the best that we have ever touched, and this is straight from the machine without doing any optimization or anything, actually is the most different of all. Which, in this case, for example, the ones that we described in one of the recent publications, they go, 20, 30% different. So they are quite different. And actually, in the retrotransposon domain, we have done similar procedures. And actually, we can probably even go more distant now that probably have a more streamlined pipeline. So, and I think here, it's not so much probably now, these are not by any means the limits of what we can explore. Here, I think, is still that we are limited in the descriptors, in these oracles. These oracles that we have, still they are pretty basic. We have, good oracles for a structure. We have some good guesses on certain important residues. But here, I think we need to generate more data and more knowledge to improve these oracles and then be able to push these frontiers more and more far.
Yeah, transposons are a great technology. I think many people in the Church Lab use them. They're excellent to deliver, very efficient, and great as a research tool and some potential as a clinical tool. Are you familiar with the Poseida clinical trial? They used piggyback transposon and for a CAR T product. But unfortunately, multiple patients got T-cell lymphomas out of this. And it does seem that your technology to not randomly insert with the piggyback, but target it. That approach should largely alleviate that. If you pick the site with intention and with certain safety testing, you can have so-called safe harbor sites that can tolerate those insertions. Could you comment a little bit about how you pick which sites to target with your targeted approach?
Yeah, I think that's an important point. It's true that as of today, it's still relatively early days in these advanced therapies, but I think probably in the future therapies, I think it's important to it would be desirable to have as much control as possible of all the variables. And of course, one of the big variables is where to insert a payload. And inserting the payload is of course a safety issue because we could activate an oncogene or other issues, but also even I think it's important for performance. For example, in our case, we have a T-cell program. And for example, when we measure cytotoxicity of our engineered T-cells, even lower level of engineering in T-cells offer much better performance in targeted than if we have higher activity, but with random. So somehow this homogeneity of the product of having all the insertions in the same position, not only we believe it increases the safety, but also it increases the performance. Yeah. And then in terms of the insertion site, so far we have been using, let's say, more famous loci that are used, for example, in T-cell engineering, like the TCR receptor. But I think here again is a part that I think there is a lot of science to be done. So for example, we have done a collaboration with a lab here in Barcelona that they are experts in chromatin structure. And I think probably a lot can be done to improve this because there are lots of interesting sites in the genome that are not yet exploited that could offer maybe some advantages compared to the current sites that we are using. I think it's something important for both the safety and efficiency.
Yeah, excellent discussion. So you're using AI to make new transposons, new editors, and where the integration site is there any effort on what payload is inserted? I mean, you say you want gene X. What about the promoter, the regulatory elements? There's been similar concern that the right ones could improve safety or efficacy as well.
Yeah, in this case, for example, the technology that we have been working on, which we call it FICAT, that combines this DNA transposase piggyback-like plus CRISPR. It performs, it scales well with size. So, and this is something that we're very careful when we're doing the development to keep this somehow capacity of inserting large payloads, where it's we believe that this is important. Again, for these T-cell therapies of the future, probably the car is not enough. So probably we need to get some immunostimulant, maybe some safety switch. So we need several elements that sometimes in some technologies we cannot fit them. For example, in a viral technology or some technology that doesn't scale well with size, like for example, a molecular combination. So here we have, have had an important emphasis for multifunctional car payloads.
Yeah, and around FICAT and these transposon technologies, you created and founded a company, Integrate Therapeutics. Could you describe a little bit about what the goal of that company was and what you're up to today?
Yes. So that was actually probably this emerged as like for the very first experiment that we did in the Symbio lab in Barcelona, and somehow once we saw that this technology started to work and also started to be quite portable in different cell types, so we thought it would be very interesting to try to push this outside of the lab. And we founded this company, Integrate Therapeutics, together with one of the postdocs that was developing this technology, Adencia Sanchez Mejias, who is now the CEO. And basically we raised some money from both local and international investors. And somehow we started very strong on building a foundry, so that then we could make this technology robust, that could produce good protocols for several important cell types like T cells or, for example, or liver. And right now, we have a good team in the Children's Hospital of Barcelona in Sant Joan de Déu, where they are trying to push this towards the patient. So especially very much focused on cell engineering, targeting this large payload type of domain, which I think is important for T-cell therapies because of this multifunctionality, but even for some gene therapies. There are some gene therapies that basically some large gene are affected. And it's true that many times we can do gene editing to correct the mutation, but there are some diseases that the number of patients is very small. And then having kind of a universal solution, we believe that in some cases it could be beneficial. So we have been having this team pushing very hard and trying to bring this to patients. It has been hard times for gene writing, but I think now we are in good shape and with very, very good results.
Yeah, I'm glad you segued to that. Wanted to talk about both the difficulties for companies like this, but also the great promise. Many companies like Tome, Scribe, Tessera having huge problems, shutting down, laying off big parts of their staff. It seems it's still difficult to commercialize on it and make money and actually drive these to therapies. But at the same time, individualized gene editing approaches, these have such promise with baby KJ and others that are advancing to make this a platform for different mutations. So it does seem there's great promise, but especially for insertion companies, it's been a difficult time. So what's your assessment of this, and what do you think the field needs to grow, to mature, to attract investors?
Yeah, so I completely agree. It has been hard times. So even companies that they had extraordinary technologies, they had some difficulties to keep going. In our case, the strategy was always to keep very organic growth. So we grew actually relatively slowly, but very robustly. So we closed the first seed, which was relatively small, in the order of 6 million. Then we closed like a pre-Series A recently, about 11 million. So it was kind of a growth, constant growth, but very, let's say, contained and with very careful planning of what we were doing. I personally think that the need of insertion technologies is high. I think it's really a strategic technology. For example, the cell therapies of the future, I think all will require some important gene writing technologies. So we are all thinking of genetic circuits that normally are complex, that they have several components. And I think probably this will just get bigger, because at the end we want the perfect T cell, that we have a Q switch, that we have immunostimulation, that we can target that place, that we can regulate these T cell actions. And all of that, I think there is, basically you need to encode it into the genome. So I think it's a really strategic technology that, I think it's worth developing. But there has been a climate in biotech, I would say even in general, that some years have been hard for multiple reasons. I don't know if it's partially competition with AI, periods of high interest rates. But in our case, the strategy has been to grow robustly, not very fast, but on a solid way, and very carefully planning the clinical program. And for example, in these last years, we have expanded very much the part of cell engineering, which is where we see a lot of value creation at this point.
And Integra, you started on focused on getting a trial in humans, or are you using the technology to partner with others to drive it? You've mentioned T-cell, CAR T therapy. Is that your clinical indication you're going after, or could you describe what the product would look like for the company?
Yeah, definitely. Here, so we really need to recognize what is our DNA. Let's say the metaphoric DNA. We are technology people. So where we kind of add value is in technology. So it's true that probably we are doing some internal programs, but we have important collaborations with companies. For example, in the case of car domain, that they have IP on cars. So we are very much partnering, especially in these indications. With others, so that then we can have the synergy of the technology and the synergy of the final target. So yeah, we are still in preclinical phase. So we have a relatively advanced preclinical phase. So yeah, we are not yet a clinical company.
Okay, excellent. And touching back to George's influence, one thing I always loved was he put this slide up of enhancements that he would want. Some are point mutations, some are knockouts, but many are actual genes that are added in. Do you think of any of this? What a great inserter gene writer could do in the far future for potential enhancement?
Well, I think that's a very interesting point. Actually, I think it was one of the first episodes. I think Matter Bio, Chris was mentioning that they are doing some large insertions for basically DNA protection purposes. So I think these are great applications. So we haven't started with those. We have started with more medical profiles, but I definitely see the value and the potential, because especially when we need to alter systemic properties, for example, in this case, enhancing radiation protection, it's very likely that it will be beneficial to have a more complex genetic circuit that probably will require multiple components, that it will be difficult to deliver with an AAV, that we need maybe something more, let's say, with a higher capacity. On the other example is genotransplantation, that is so close to my heart, that I have worked so many years in genotransplantation. There, we have massive payloads. We are delivering sometimes 10 genes or 7, always a big number of genes. So I think some of these frontier applications will definitely require this capacity to do this kind of more, let's say, radical engineering of the human or animal genome.
Excellent. And I'm glad you brought up eGenesis. We've mentioned it briefly, but maybe you could take a couple minutes to tell the audience about this exciting program to bring pig organs into the human. And they've had very exciting clinical results lately in human trials.
Yeah, I think it's one of the, I always use it as one of the most fantastic examples of extreme synthetic biology, right? And come true. And actually, I remember when I was working in xenotransplantation at eGenesis, we were going to conferences, and there were people doing a joke that is very unpleasant. They were saying, xenotransplantation is tomorrow, and it will always be, no? Or xenotransplantation is the future, and it will always be, and ha ha ha. So definitely, this job cannot be done anymore, right? So something that sounds so complicated, basically, I think it had very encouraging results. And here again is really genetic engineering brought to the extreme. Well, I don't know if to the extreme, but actually to high levels. So basically, these Genesis pigs have maybe 50 modifications, combining many different types of modifications, right? Lots of knockouts, knock-ins. So it's and it's, I think, and it's all a pre-AI kind of development. So here, with the inspiration of biology, of, let's say, looking what do the pig has that we don't have, then we take it out. What is key from our genome, like, for example, altering coagulation, altering certain parts of the immune system we can add. And with that, we can really do these transformative changes. And really, basically, I think it's really a dream, right? That we can create this unlimited, completely unlimited supply of organs for transplantation. Because this is of course, it's very hard to build this two-dimensional system that has these advanced functionalities like a kidney. So I think it's a very inspiring example of basically what incredible things synthetic biology can do.
And those who doubted xenotransplantation, who made the joke, as you mentioned, they broke George's rule, right? They're saying it's impossible. That's another way to say it, but always tomorrow is another way of saying impossible, and that many talented engineers just take that as a challenge. And I think it turns out it's very risky to say something's impossible. And I really like that rule of George's, and he'll always call you out for it if anyone says that.
Yeah, definitely, especially in biology, because we have seen so many extreme properties in the natural biosphere. So I think it's very hard to, to basically say that something will never happen.
Yeah, excellent. So we talked about organ transplantation, some of your work with transposons. I'm interested in that third pillar you defined of the skin microbiome. With these approaches, are you engineering the bacteria or just finding different natural strains that can alter a certain, as you mentioned, acne or other traits?
Actually, both. So we started first just demonstrating, basic technology demonstration that we could change the skin microbiome. And this we just published, made the first patents. And actually here, the key was the very special anatomy that the bacteria that we use basically occupies. It occupies the sebaceous niche, which is, because our skin is not flat, has all these appendixes that basically where the sebaceous gland is. And somehow this part has some properties that are very interesting. First, it's very stable. So if we put something there, it stays there, which is interesting for engineering long-term properties. And second, the skin is kind of a desert, right? The skin isolates us from outside, but not in these deeper parts. Actually, when we use a topical medicine, normally 99% enters through these appendixes. So it's a very interesting area to do synthetic biology on. And in the beginning, once we could demonstrate that we can engraft these probiotic solutions, then we formulated several of these probiotic solutions with the aim to create a probiotic against acne. And here the reasoning was very simple. So there are some strains that are very associated with acne because they have a strong inflammatory profile. And there are some others that are completely the opposite, the same as E. coli, right? That you have E. coli Nisl, which is very beneficial for the gut, and then you have some E. coli that could kill you in a few days because... And here, so basically by just this very simple concept of like selecting certain biomarkers and then preparing these solutions, basically we did several clinical testing. It worked quite well in the framework of this company. It's Biomedic that we created many years ago, 10 years ago or more. And this eventually finished quite well. So it was at the end acquired by Weisdorf, and they are continuing the development of this. And I think it will generate a very interesting product. However, in our lab, we kept being very fascinated by this bacteria, but in this case, more as a synthetic biology chassis. At the end, if it's a bacteria that occupies a site that is interesting for physiology and that is stable, that's fantastic. So it's what we want. So now, we are building different genetic circuits to perform more specific actions. In this case, the actions that we have explored is sensing. So we have been making sensors for temperature. Also, we publish a sensor for inflammation and also other things. And then also circuits to do what we call actuation, which is, let's say, perturbing certain properties. For example, we wanted to imitate Accutane. Accutane is a typical acne drug, but it has a difficult safety profile. So here we wanted to create a bacteria that does a similar effect, that stops sebum secretion. And in this case, we create a genetic circuit that in this case stops the sebum secretion. So somehow, we created, we are using C. acnes as a chassis for the skin, and we have been tinkering with different properties. Also now we are expanding more on the inflammatory, let's say, perturbation, exploring different pathways from indole pathway, ROS, or even some specific cytokines. So I think it's an interesting system. I think it was quite well proven as a natural probiotic, but we believe that also it has very interesting, more, let's say, defined applications in this more, let's say, GMO label style.
Excellent description. And next week, we're actually getting Jonathan Scheiman on with Fit Biomics to talk about his Nella and V-Nella products originally derived from pro athletes, but actually have some pretty good clinical data that they're developing that it's beneficial for not just performance, but for sleep health, for GI issues, for autoimmune issues. It's a very exciting stuff to manipulate the microbiome. And
I agree. Because these bacteria, they have co-evolved with us for so many thousands of years that actually somehow we established a very solid molecular development, like a molecular dialogue with them for epithelial development, immune system, metabolic functions. And it's surprising. For example, in the skin, we get surprised almost every month with a new function that we could not predict that somehow these microbes can influence, like from immune system, sebum secretion, and even some things connected to the neural well-being. So it's yeah, it's very curious how much is being uncovered and how powerful it is to use these microbes for different health interventions.
It seems like a great target for actually engineering those microbes as opposed to the body. Much harder to reset or do any changes, but if anything happens with a engineered bacteria, you can always clear the area quite well and recolonize. Has risks, but what type of modifications might you want? Like what type of enhancements? Or you mentioned Accutane-like function for acne. Are there any other targets that you're going for that would give these bacteria a more beneficial profile?
Yeah. For now, we have been, let's say, very medically oriented. So we have worked a lot on acne. So we created these different strains, both natural, which work very well, and we have now in advanced preclinical testing these non-natural ones that they are a bit more radical in the effect. Also, we have been very interested in atopic dermatitis, so somehow sensing, so to teach the bacteria how to somehow sense our inflammation and then being able to deploy. Because at the end, here the inspiration was, at the end here is not only a delivery mechanism. This should be somehow some tool that should allow us to control spatially and temporally how to administer this drug. Since bacteria can do that, they are a very sophisticated molecular machine. Then here the concept was to try to couple the sensing with some kind of actuation machinery so that basically we can get this initial action just after it happens. So this is the vision. But I think there are other very interesting applications. So we are collaborating with Tysilk Moon here in the US, in the JCVI. For example, even engineering a bit more crazy things, like, for example, using these microbes to generate heat. So there is a lot of chemical energy in the skin. So trying to somehow capitalize this chemical energy to transform them into heat. Or even other things, like, for example, mosquito repellent. I think that's also another interesting option that these long-term interventions could have an important impact. So so far more medical, but I think we will open up to other things.
Yeah, I'll have to look into that research with Tysilk Moon and the JCVI. With heat, definitely could see it for, many people just want to be warmer, but also space travel potentially or other extreme environments. That could be very useful. And obviously mosquito repellent, that is such a big impact with malaria and others. That could be hugely beneficial. So interested in following that up. And this leads to the next topic of how do you balance ethics and safety with these new tools? It's great to think of these ideas. Many have an immediate potential upside, but especially with AI-designed proteins, how do you put in correct guardrails and safety protocols to make sure that we're proceeding ethically?
Yeah, I think this, of course, it's always difficult, right? We are clearly undergoing exponential times, right, in many different channels, sequencing, writing, editing, and now speaking. So, of course, this is it could have potential dual-use technologies, and biology, by nature, has certain complexities, right? First, after COVID, no one can deny the destructive potential of biology, right? It was very clear how many millions of deaths by COVID. And second, biology is a decentralized technology, so it's very different. So basically the strategies that work for nuclear energy, I don't think are very promising for biology, because basically, first, it's decentralized, and second, basically is not material intense. So here, I think we need to be very, basically, more creative. And I think here, probably, I've seen different strategies. I think surveillance is an important one that I think many people have put on the table. Access to synthesis, I think it's a very important filter to regulate what we want to enable or not. As of today, DNA synthesis is quite centralized, not completely, because there are companies that can sell you a synthesizer, but it's hyper centralized. And then, and of course, research materials. Of course, there are systems that are more risky than others. And this, I think we need to be very vigilant and try to be as proactive as possible. Because clearly, all these things that excite us so much about these incredible properties of biology, it is undeniable that they could be used with, let's say, not very nice purposes. So, in my case, I have been very engaged in this type of things. So I participated in analysis groups of risk in biotechnology in the European Union. Also, I participated in other debates in private foundations. It's a topic that I'm very, very interested and definitely, and also other people on my lab. So I think it's very important to coordinate this technology development with being cautious in the steps that we take.
Excellent. And one great thing about your background is you've jumped around a lot, had the European point of view, the American point of view. What have you learned about the different ways people think across the world and how you can synthesize that?
Yeah, I think it has been beautiful to be exposed to these different, let's say, cultures. I have extremely good memories from Boston. I always say, like, being in Boston for a scientist is like a kid in a candy store, right? So it's a very, very unique place. And definitely from America, I brought with me this entrepreneurial kind of mentality, this no fear for risk, right? Working with agencies like, I know, DARPA, like all this huge groundbreaking, and also this very mature ecosystem, right? That you have strong private sector, strong academic sector, strong hospitals. I think this is really way more than the sum of its parts. In Europe, I think we are doing well in what is basic science. So I think Europe has been very competitive in basic science. Probably we need to get better in translating the science into useful outcomes, which definitely here we are behind America and also China. And recently also have traveled to China. We have very good friends like Lu Hanyang. And I am very impressed also the developments in China, for example, this massive investment in infrastructure, talent recruitment. For me, it has been a pleasure. And I try to interact a lot, have collaborations in America. Also, I interact a lot with Asia. So this is one of the things that I truly like from science. I would still like to think of a science as something quite universal, beyond culture, beyond... I know of the complexities of the geopolitical situations and so on. But I personally have enjoyed a lot of these, let's say, cultural exchanges. And I try to cultivate them and also transmit them to whatever, like my students and postdocs.
Yeah, I think science can really bring people together to work on a shared problem, and that's really a beautiful thing. And we've talked about AI more as the mind, predicting, designing new things. Does your lab, or what are your thoughts on use of AI towards automation? So you actually have to test these in reality, and there's been huge advances in the liquid handlers and robots that can execute these experiments. And what do you think about the current status of that and where it's going?
Yeah, here I think it's a very important part, because I think we really need to upgrade our throughput in experimental side. So I know that many companies have made a lot of progress, Ginkgo and others. Also, I was very impressed in China in the recent visit, how much investment in infrastructure for high throughput was made in different parts of the country. In my case, also, we have been working quite a lot. Of course, we are a lab, so we're more modest kind of project. But yeah, we are trying to bring new concepts. So for example, we have been building something that we call the synthetic evolution machine. And here we try to really build a paradigm where basically the numbers that AI needs are not longer constrained by the hardware system. And here, basically, the best way that we found to implement that is using DNA. So basically having an agent that can, let's say, generate and learn, kind of a computer server. And then we have what we call basically some kind of robot that executes these changes. And this robot basically takes in DNA and outputs DNA. So DNA as an encoding and DNA as a encoding of the phenotype. So encoding of the genotype and outputs an encoding of the phenotype. And here we just wanted to avoid the cloning, assembly, or anything like that. So it's directly DNA synthesized in chips, then basically some kind of protein barcoding technology directly into cell-free systems. So then we can still keep these thousands or tens of thousands, or even potentially hundreds of thousands of experiments simultaneously. So that then this can be read directly. So written and read by using DNA, using nanopore sequencing, using DNA synthesis in chips to the agent. Of course, it's more, let's say, proof of concept, because we don't have the capacity to measure a lot of phenotypes just with DNA. Here we are limited to protein-DNA interactions, protein-protein interactions, but we really wanted to somehow put these things together and see if we can truly compute on DNA. And here, of course, one part of the agent is server, classical computer, but the other part is a wet computer. It's slower, the wet computer, but we try to make it as fast as possible. That can do, let's say, the computation of these hundreds of thousands in a day. So So that's the goal and the vision, and I think it's a very important problem. It sounds, maybe engineering, but not only engineering. I think if we want to learn more about these cell models that we were mentioning before, if we want to get better oracles, it's completely essential to start increasing the throughput so that then we can match, right, the needs of learning, of deep learning of these algorithms. So, yeah, I think it's a very important problem that we try to make our modest contribution.
Yeah, it's excellent. I'll dive into that more. And I'm glad you brought up AI agents. So one thing that's taking over the internet is Claudebot. Not sure if you've read into this or implemented it, but have you or anyone in your lab started implementing any of this towards the science that you're doing? I think from what I've seen, it adds a great capability to run your agent locally and then have a lot of preemptive reports, like a daily report or constant scanning as opposed to reactive to the specific prompts that you're doing. Do you see any potential of this for your science or your work?
Yeah, I am personally. So I haven't. I don't have any programmed agent for this more, let's say, knowledge-based AI. Even though I see very much the point, and I think it's something very interesting. But I am a very strong user. Every day I use Gemini a lot. I use the Edison machine from Public House a lot. So, and this complement very much our work. So we try to not only search but also cross with our data, our internal nodes. And yeah, I think it's at least every day I dedicate, if not an hour or some more, talking with some kind of Gemini or Edison machine. And I enjoy very much these conversations because biology is very chaotic, right? It's a body of knowledge that is very unstructured, especially when we go to concepts and regulatory layers. And I think getting better in using these tools, not only prompts, but also learning how to use the right mathematics to analyze this, let's say, network of knowledge. I think that these are things that probably will bring us closer to this oracle of a full cell or of a full organism. So, yeah, and maybe we'll have even to rethink how we write papers. Maybe we'll have to do some, let's say, sophisticated processing of how we teach, basically how we report the results. But yeah, I think it's very exciting times because the tip of the iceberg is already very promising.
It's amazing what these tools can do, and talk with them and have to accept their limitations, certainly, especially for science and hallucinated references or hallucinated ideas. But as a broad research tool, it's amazing what they can do. But one limitation I saw with Gemini, Claude, others was long-term memory is not that good. It could remember certain aspects, but that's what I really like about Claudebot, and I've been setting up a researcher that it really keeps a complete, comprehensive MD file of everything that you've talked with it about. And I'm trying to build that up, and that's one of my goals I'm implementing, to build an AI researcher scientist that fully understands all my goals, all my missions, and get it to be automated and watch it grow as the technology grows.
No, I definitely think I'm very curious to learn more about this, so we'll definitely check it out. And actually, recently I had a conversation with a pharma executive, and I was quite impressed also how they use this in, let's say, this more industrial side connected to life sciences. So, of course, just the fact of having an LLM that was somehow crossing all their reports of the clinical testing, the regulatory, the meeting notes with experts, he was telling me that now he can do so much more crossing of things. And I said, well, of course, this is transformative, right? This access to knowledge, this access to, crossing information, which, I think it's very worth spending time to make use of it.
Yeah, at one point of that, definitely I think exciting potential for regulatory documents for that type of submission. A potential question I've heard asked by the FDA is being prepared to have an answer of how you implemented AI in the process. Like, which prompts did you use? What records did you do? Which parts of this were AI generated? Which parts were edited? And I think most people don't have a full record. They might look at their conversation history, but thinking about how we present our AI strategy, both for papers, for grants, or for regulatory documents, I think will be important to be transparent in the future so you can be audited.
Definitely, and always validating with, basically experimental data or direct references, anything that came up, right? Yeah, definitely, I completely agree.
Yep. All right, we're coming up to the end, but I always like to talk a little about science fiction. Could you describe how or if science fiction has influenced your science, your view on the world, or your work?
Yeah, actually, it has been quite influential, and I think from different points of view. I think first, I think it's very clear that science fiction has been a better predictor of science than scientifics ourselves, because I think we are too skeptic. So, so, and I think many examples of this, right? From I don't know, probably many people when they saw Star Wars, they wouldn't think that we can have a robot that has cognitive properties similar to humans. Now it's not surprising, right? I'm sure we'll have soon how it's called, well, some kind of robot, an Optimus or something that will maybe help taking care of our kids or home or whatever. And then another example also is Jurassic Park. At the end, for many years, when we have been so DNA centric, right? We thought this is impossible, right? This is just a fantasy. But bro, so now we have AI that can convert, let's say, images to text. So I don't know, can we convert 3D images to DNA? Maybe it doesn't sound that crazy. And after, I don't know, you mentioned colossal before, right? I don't think it sounds impossible that we can really make incredible things using, basically large impact phenotypes, synthetic genome prediction. So so I think it's interesting, and also I think we need to keep our mind very open. These are maybe more practical things, right? That we have seen that probably science, at least for me, has surprised me, right? AI has surprised me. Also, maybe the capacity to bring back phenotypes also something that has surprised me. But also, I think even we need to be quite, I don't know, open-minded about what is life, right? Which I think science fiction has been way more open than us in what is life. So it's very difficult to predict what is life, right? Now we have seen one example of life, which is really a cosmic aberration. It's something that is very different from the rest of things that we found in the universe, which is based on DNA. It's beautiful, right? We are so inspired, all of us, with it. But predicting what is life is very difficult. Actually, mathematicians would tell you that it's impossible, right? It's very similar to the halting problem, right? It's basically like what Turing said, right? It's really impossible to say, right? I'm sure we can find cues, right? People thinking about this assembly theory, right? Of like maybe detecting peaks in complexity, and complexity probably something that must be an ingredient of this, of something that perpetuates its existence, like in our case DNA, right? That we have had these different inflection points on order, right? Let's say from whatever, plasma to molecules and then molecules to life. This was a big jump in complexity. And now technology has been another big jump of complexity. Right? At least we had these two big inflection points. One, from molecules to life, and now from, let's say, reality to technology to document this reality and create very structured examples of reality. And AI, of course, being a big, pusher of this complexity classified as technology. So I think it's very important to read these books and not to limit ourselves with the papers that we have published yesterday or something like that. It's, like, I don't know, things like the story of Asimov, right? The last question brings me back so much, this image, right, of this versus in complexity that we are experiencing. So I don't know. I think it's very important to cultivate this. And I think fiction is very interesting to, set free our imagination. I don't know. I've read some fiction. For example, some recent book that I really like is actually a book that was initially written in Spanish. It also exists in English, which is called Maniac by Labatut, which is a curious mix of history and fiction, and is mostly centered on John von Neumann life and the chaos of the 20th century physics and the emergence of artificial intelligence. I think it's a book that I enjoyed very much, also because of the topic and how it's written. Another book that I really like, that I read maybe 1 or 2 years ago, 1 year and a half ago, is The Observer by Victor Lanza, which is kind of is fiction, but is basically based on his theories of biocentrism, of how mind connects to reality. I had a lot of fun reading it, right? Basically, potentially engineering the brain could be used to travel across parallel universes. So I think it's an interesting mental exercise. And So yeah, I think it's definitely with my kids. I love sharing with them as much as I can on this.
Yeah, thanks for sharing those recommendations, and I love it that the skeptic in the scientist could keep us back at times. It's critical for any individual experiment, but for that bigger vision of what's possible, like in Star Trek, the doors opened automatically. So many of these things were just put into reality, called forth well before we had any technical means, but as soon as people saw it, then they can build the engineering path. So it's very inspiring for that. Well, we're at time, Mark. Thank you for coming. I really appreciate you sharing your work and your progress with it. This is really great, rich history of the companies that you've started and the very exciting science and the philosophy you really approach to understanding life and biology. So thank you, and have a great day.
Oh, it was fantastic. So I really enjoyed the conversation.